In this video, we are going to discuss the questions on Chapter 7, Gaseous Exchange and its Control. 1a. Figure 1a shows a structure of a hemoglobin found in human erythrocyte, which is the red blood cell. Identify structure labeled X and state its function. Make sure you answer the questions accordingly. You have to first answer structure X and then only its function. Based on the picture here, X is the heme group. There are two spellings, either HAEM or you can spell it as HEME. And the function of heme group is to bind with oxygen or to act as the oxygen binding site. The second question, give two characteristics of hemoglobin molecule that enable it to transport oxygen effectively. This is all dependent on the affinity of the hemoglobin to oxygen. So, the affinity of hemoglobin to oxygen changes based on the partial pressure of oxygen. When the partial pressure of oxygen is high, which means we have a lot of oxygen, then the hemoglobin will have high affinity to the oxygen, which means it will bind easily to oxygen. Whereas when the partial pressure of oxygen is low, that means it will have less oxygen. In this case, the hemoglobin will have low affinity to oxygen, which means it will release the oxygen easily. And this also shows allosteric cooperativity. Number 3. What is the maximum number of oxygen molecules that can be carried by one hemoglobin at any one time? One hemoglobin consists of 4 polypeptide chains, that's why 4 heme groups, as you can see in this picture. 1, 2, 3, 4. 4 heme groups, that's why it can bind maximum 4 oxygen molecules. So the answer for number 3 is 4. Let's look at the second question here. Figure 2b shows a way of carbon dioxide transport in the blood. 1. Name the types of carbon dioxide transport in figure 2b, and that's the first part of the question, and then you have to state its percentage. We know that there are 3 ways of carbon dioxide transport in the blood. The way that carbon dioxide is being transported in this picture, you can see from this clue here, is as bicarbonate ions, and the percentage is 70%. 2. What is enzyme A in figure 2b? In the diagram, enzyme A is involved in catalyzing the reaction between water and carbon dioxide to form Y. Enzyme A is simply carbonic anhydrase. Number 3. Identify Y and Z in figure 2b. So carbon dioxide combined with water, it will form H2, 1C, and then 3O. So H2CO3, or we also call it as carbonic acid. So in the exam, you can either write H2CO3 or carbonic acid. And this carbonic acid, it is a weak acid. That's why it will dissociate into bicarbonate ions as well as Z, which in this case, H+. Because 2H, now this 1H is in the bicarbonate ion, so another one is the hydrogen ion. And the last question here, Z will then bind with hemoglobin to form X. What is X? So that's the first part of this question, and then you have to tell the importance of X. Having hydrogen ions in the red blood cell will lower the pH in the red blood cell. That's why we need to have the hydrogen ion combined with hemoglobin, and that will form HHB, which its full name is hemoglobinic acid. The importance of forming hemoglobinic acid is to prevent the decrease in pH in the red blood cell. So we don't want the red blood cell to become acidic, to prevent the red blood cell from becoming acidic. That's the importance of the HHB. The second question here, figure 3a shows the oxygen dissociation curves P and Q of two different pigments involved in the transport of oxygen in mammals. State the correct label for the following. Which curves represent the oxygen dissociation curve of hemoglobin, and which one represents that of myoglobin? In this case, we will have P represents the curve for myoglobin, whereas Q is the one that represents the oxygen dissociation curve for hemoglobin. How do we know? You can tell from the position of the curve, which myoglobin is to the left and hemoglobin is on the right. Or you can tell from the shape of the curve. If myoglobin, the curve is hyperbolic shape, which is this shape here, straight and then curve and then straight. What about hemoglobin? The oxygen dissociation curve for hemoglobin is sigmoid shaped, which is S-shaped. So you can see here, and that's the hemoglobin. 2. Explain briefly the difference between the oxygen dissociation curves of hemoglobin and myoglobin. So the first one, we compare the position of the curve. The oxygen dissociation curve of myoglobin is located on the left of the oxygen dissociation curve of hemoglobin. We always relate the curve on the left to higher affinity for oxygen. You can tell from this curve. Let's say we have the partial pressure of oxygen of 20, and then when we look at the graph, you can see that hemoglobin will have a much lower oxygen saturation compared to myoglobin, which have about 70% oxygen saturation, which means that at this low partial pressure of oxygen, more myoglobin contains oxygen compared to hemoglobin, which tells us that myoglobin has higher affinity for oxygen, which means it binds more oxygen compared to hemoglobin. And myoglobin will only release the oxygen at very low partial pressure of oxygen. The function of myoglobin is to store oxygen in the muscle. That's why it will not release oxygen easily. It will only release the oxygen when the partial pressure of oxygen is very low. That's why we say it has a very high affinity for oxygen compared to hemoglobin. B. Figure 3b shows the oxygen dissociation curves of human hemoglobin under different partial pressure of carbon dioxide. According to figure 3b, arrange the oxygen dissociation curves in ascending order of partial pressure of carbon dioxide. Ascending order, which means from low to high partial pressure of carbon dioxide. So the arrangement will be A, B, C. Low partial pressure of carbon dioxide, which means that it is higher pH. When the pH is higher, the curve will be higher, and that will be to the left. So A, the curve A being on the left, it tells that it has the lowest partial pressure of carbon dioxide. B being in the middle, so it has average level of partial pressure of carbon dioxide, and then C being the lowest curve That means it has the most partial pressure of carbon dioxide, the highest partial pressure of carbon dioxide. That's why it has lower pH. When it has lower pH, the curve will also be lower, which is on the right, as shown in the diagram. According to figure 3b, name the effect and explain how could this happen. So the first part, you must mention the name of the effect, and then only you tell what caused this effect. This is what we call the Bohr effect, or we can also call it as Bohr shift, which tells us that increase in partial pressure of carbon dioxide will lower the blood pH. How can this happen? You learn about the transport of carbon dioxide. We know that carbon dioxide can combine with water in red blood cell to form carbonic acid, H2CO3. And this carbonic acid will dissociate into H plus and HCO3 minus. What happens when the concentration or partial pressure of carbon dioxide increases is that it will also increase the hydrogen ion concentration, which then lower the pH in the blood. And this is what we call the Bohr effect. Increase in partial pressure of carbon dioxide, lower the blood pH. And what will this cause? It will result in a decrease in the affinity of hemoglobin for oxygen, which means hemoglobin will release oxygen easily because the affinity for oxygen is lowered. The curve shifts to the right. And that's what we call the Bohr effect or Bohr shift. Number 3. 5 teenagers died from carbon monoxide poisoning after they left their car running in the garage beneath their room. Explain how carbon monoxide causes poisoning. First, you have to know that hemoglobin, it has higher affinity for carbon monoxide than oxygen, which means that hemoglobin will bind carbon monoxide more easily than to oxygen. When in the presence of carbon monoxide, hemoglobin will choose to bind with carbon monoxide than to oxygen. This is what it means by higher affinity for carbon monoxide. So it will choose carbon monoxide over oxygen. It can bind easily with carbon monoxide than to oxygen. And that's why less oxygen bind to the hemoglobin. When we have less oxygen bind to the hemoglobin, it tells us that it will have less oxygen being sent to the body. And what will that cause? It will cause oxygen deprivation, or we say oxygen starvation throughout the body, which means we lack of oxygen in the tissue. And this is all because hemoglobin binds to carbon monoxide instead of binding to oxygen. So we don't have enough oxygen carried by the hemoglobin to the tissue, and this will cause oxygen deprivation. This part we are going to learn the assay. So let's fill in the blank. Describe the transport of carbon dioxide from tissues to the lungs in human. Carbon dioxide can be transported in 3 ways, like I mentioned just now. Firstly, as the carbon dioxide dissolves in the blood plasma. Secondly, it can combine with the amino group of the hemoglobin. You should remember that carbon dioxide combines with the amino group, while oxygen binds to the heme group of hemoglobin. There are 2 different parts in the hemoglobin. Carbon dioxide binds with amino group of hemoglobin to form carbaminohemoglobin. From the name itself, you can tell it is the amino group of the hemoglobin that it is bound to. Lastly, majority of the carbon dioxide, which is about 70% of the carbon dioxide, is transported in the body in the form of bicarbonate ions. If you don't want to spell it out, you can just write HCO3 minus. In respiring tissue, the partial pressure of carbon dioxide is higher. Of course, in the respiring tissue, which means the tissue that carry out cellular respiration. You learn that during cellular respiration, glucose is being broken down to form carbon dioxide and then water and ATP. So we have carbon dioxide produced during cellular respiration, and that's why in the respiring tissue, the partial pressure of carbon dioxide is higher than that in the blood capillary. Therefore, carbon dioxide will diffuse into the blood capillary from the respiring tissue, and then only it enter the red blood cell. You can either write red blood cell or erythrocyte. Both are acceptable. And then this carbon dioxide will combine with water in the red blood cell to form carbonic acid. Or if you don't want, you can write H2CO3. This reaction is catalyzed by the enzyme carbonic anhydrase. The acid form, which refers to this carbonic acid over here, will then dissociate into bicarbonate ion. Again, you can just write HCO3 minus and the hydrogen ion, which is H plus. The bicarbonate ion HCO3 minus will diffuse out of the red blood cell. This mass movement of anion means the negative charge ions, out of the red blood cell causes the cytoplasm of the cell to become relatively positively charged. And that's why we will have the chloride ion to diffuse into the red blood cell. This is a process that we call the chloride shift. And what is the importance of doing chloride shift? It is to maintain the electrochemical neutrality, or we say the electrochemical balance of erythrocyte. The hydrogen ion produced from dissociation of carbonic acid H2CO3, it will bind to hemoglobin. So like I mentioned just now, it will combine with hemoglobin to form hemoglobinic acid. Or if you don't want, you can just write HB. HB stands for hemoglobin, H stands for the hydrogen ions. And this is to prevent the decrease in pH in the red blood cell. So in this case, we say that hemoglobin acts as a buffer, a pH buffer. It is to neutralize the acidity of the red blood cell. Just now, we were talking about respiring tissue, and now it is in the alveoli of the lungs. The partial pressure of oxygen, of course, it will be higher because we breathe in the oxygen and it's at the lungs. So the lungs will have the highest partial pressure of oxygen than anywhere else in the body. So the partial pressure of oxygen is higher than that in the blood capillary. So the hemoglobinic acid, the one that we mentioned just now, and again you can just write HB, will dissociate to form hydrogen ion and hemoglobin, so that the hemoglobin can bind to oxygen, because now the partial pressure of oxygen is high. So the hemoglobin is ready to bind with the oxygen. At the same time, the partial pressure of carbon dioxide in the blood is higher than that of the alveoli. the bicarbonate ion, or we say the HCO3 minus ion, will diffuse back into the red blood cell and combine with the hydrogen ion from the dissociation, wrong spelling, of hemoglobinic acid to become carbonic acid. Everything is reversed. Just now, carbonic acid dissociate to become hydrogen ions and bicarbonate ions. Now everything reversed. From hydrogen ion and bicarbonate ion, they will combine to form back the carbonic acid, or H2CO3. And because bicarbonate ions already enter back into the red blood cell, so chloride ion will need to diffuse out of the red blood cell so that it can balance the charges inside the red blood cell. The acid form, which in this case the carbonic acid, will dissociate to form carbon dioxide as well as water again. They will convert back to carbon dioxide and water, and then this reaction again is catalyzed by the enzyme carbonic anhydrase. We use the same enzyme to catalyze the forward and the backward reaction. Carbon dioxide will then diffuse out of the red blood cell and then out of the blood capillary into the alveoli, where it will then be exhaled, so we breathe out carbon dioxide. Let's look at the regulation of breathing in human. Breathing is controlled by respiratory center in the part of the brain that we call medulla oblongata. The respiratory center in medulla oblongata can be divided into two. One that we call the inspiratory center. From the name, you know it controls inspiration, which means inhalation, breathing in. And we have another one that controls breathing out that we call the expiratory center. Accumulation of carbon dioxide due to vigorous exercise results in the decrease in the blood pH, like I mentioned before, because when we have carbon dioxide, it will combine with water to form carbonic acid. And this carbonic acid will then dissociate into H plus as well as HCO3 minus, and the increase in carbon dioxide will increase the H plus, and that decrease the blood pH due to an increase in the concentration of hydrogen ion. The increase in level of protons H plus in the blood is detected by peripheral chemoreceptors, which are located at the aortic and the carotid bodies. And some of the hydrogen ions will diffuse from the blood into the cerebrospinal fluid, which is the brain fluid. So this brain fluid, cerebrospinal fluid, will be detected by the central chemoreceptor that is located at the medulla oblongata in the brain. The various chemoreceptors, both the peripheral and the central chemoreceptor, will then send nerve impulse to the inspiratory center and stimulating it. This inspiratory center will then send nerve impulse to the effector, the first one, the diaphragm, via the phrenic nerve, from the name phrenic nerve. And another effector involved is the external intercostal muscle. And how to send the nerve impulse to the external intercostal muscle is via the thoracic nerve, or sometimes we call this as the intercostal nerve. And then causing both the diaphragm and the external intercostal muscle to contract. The contraction of these muscles will then increase the volume of the thoracic cavity and thus lower the pressure. When volume increases, the pressure decreases. When the volume of the thorax increases, the pressure in the thorax decreases, and that causes the atmospheric pressure to push the air into the lungs, and inhalation occurs. And when we breathe in, it causes the alveoli to expand, so our lungs expand and our lungs stretch, and this will stimulate another receptor that we call the stretch receptor on the wall of the alveoli. Hence, a message is sent from the stretch receptor. A nerve impulse will be sent from the stretch receptor to the expiratory center. And this time it requires the nerve that we call the vagus nerve. So vagus nerve will connect the stretch receptor to the brain expiratory center. And from the name itself, you can tell expiratory center, it must be something to do with exhalation. Activating the expiratory center will automatically inhibit the inspiratory center, thus stopping the stimulation of diaphragm and external intercostal muscle, and they will relax. No stimulation, so both the diaphragm and the external intercostal muscle will relax, and when they relax, the volume of the thoracic cavity will decrease. When the volume decreases, the pressure will increase. So the pressure becomes higher than that of the atmospheric pressure. The pressure inside the thorax is higher than outside. That's why the air is drawn out of the lungs. Exhalation occurs. And when this occurs, the stretch receptor is no longer stretched because we already exhale, breathe out. So the lungs not expanding anymore, is not stretched anymore. So stretch receptor is no longer stimulated. The stimulation of expiratory center will then stop, and lifting the inhibition of the inspiratory center. It's about time to start breathing in. So no more inhibition on the inspiratory center. The cycle can repeat again until the excessive carbon dioxide is expelled from the body.